So let's give some actual hard examples of ways that companies are modulating things. So let's look at the plant-derived compounds and ways that people are trying to come up with novel oral formulations. So cannabinoids don't have good bioavailability when they're delivered orally, not very much of it gets into our bloodstream. And so people are trying to come up with strategies to try and improve the bioavailability of phytocannabinoids when given orally. So to give some example, we've got some oral solutions that are being developed, and an example would be Emerald, who are making an oral CBD liquid. This would be similar to Epidiolex, which is a liquid as well, but they are claiming to have improved bioavailability, and they're looking in an autoimmune disorder called cutaneous systemic sclerosis. There are some companies making capsules like Aphios and Salt Farm. One is making a time-release CBD capsule that could be used in opioid addiction, and the other is making a gastro-resistant CBD capsule, the idea being that the CBD wouldn't be released until it got to the lower digestive system, and that would improve absorption. And they're looking at that in epilepsy, so they're trying to be a different, like a new type of Epidiolex. So having proved that CBD already works in epilepsy with an oral solution, could we do it better with a different type of delivery mechanism, such as a capsule? And there are companies who are trying to make hard-pressed tablets, and this is one of the most common form of medications, is in a hard-pressed tablet. Although there aren't many cannabinoids available in this mechanism because of the physical chemical properties of cannabinoids. But some examples would be Columbia Care, who are making a hard-pressed tablet formulation of cannabidiol or THC that has extended release, so should in theory give you higher plasma levels of these compounds for longer, which obviously is going to be beneficial for kind of more prolonged symptom management. And Echo Pharmaceuticals also have a CBD hard-pressed tablet called Avasol, which is supposed to have improved bioavailability, so get into your bloodstream better. So these are various different formats of oral formulation that companies would have IP on and would be promoting for their ability to improve drug delivery. So what about some examples of novel delivery mechanisms? How else might we be able to get cannabinoids into our body other than inhaling or through the mouth? So one example would be transdermal gels. So there are two companies, Botanix and Zenerba, who are looking at transdermal CBD gels. So this is a way that you can have CBD absorption through the skin rather than in the mouth. And they are being pursued with Botanix, they're being pursued for dermatology studies. So obviously you're directly applying the CBD to the problem area in skin disorders, which is going to be of benefit. You've got the best chance of the drug working that way. And then Zenerba have a CBD gel which they're using actually for systemic delivery of the drug, and they've looked at various disorders, but they're currently focusing on Fragile X syndrome. And they have positive data in a phase 2 study which did suggest that this transdermal CBD was effective in Fragile X syndrome. And then there are novel inhalation devices, so either inhalers or vaporizers or vape pens, so different ways of inhaling other than smoking. Can you have a more controlled, dose measured, metered mechanism of delivering specific doses of CBD? And there are some in development. So Tilray have a THC pen, and Tetra Biopharm have inhaled or vaporized products, which are various combinations of THC and CBD that they're pursuing in cancer pain. There's at least one company who is making suppositories. So this would be a rectal route of delivery of cannabinoids, which does have benefits because you can have good absorption of cannabinoids through the colon this way, and They could be, for example, useful in disorders where you want to deliver cannabinoids straight to the digestive system, such as colitis, although that has yet to be tested. But this company, Panaxia, do make rectal THC suppositories and also vaginal THC suppositories, which would be used more for endometriosis and menstrual cycle related pain. There are other companies, not pharmaceutical companies, but there are companies, for example, making CBD infused tampons for the same reason, that you could have local delivery of CBD for endometriosis and menstrual pain. And then there is, that I'm aware of, just one company who's looking at potential intravenous delivery of cannabidiol, and that's in neonatal hypoxic ischemic encephalopathy, which is basically when babies are starved of oxygen at birth, and that can lead to a number of significant neurological problems and cause cell damage at the time. And there's a lot of data that GW have done preclinically to show that IV administration of CBD after this global hypoxia, so the global starvation of oxygen, can really be neuroprotective and help stop cell death and cell damage and all of the neurological deficits that would be associated with that. And that clinical study is at least underway now, and it was in the press in the UK a couple of months ago when the first IV dose of CBD was delivered to a baby in hospital who had suffered from oxygen starvation. So these are just different ways of being able to administer cannabinoids, and it just shows, they may be superior to what we currently have, which is solutions and sublingual sprays. Any of these delivery mechanisms may help to get the drugs better into our system, which means that they have a better chance of working and we have better chance of getting positive clinical trial data with them. So something else that people are trying to do with plant-derived compounds is look at structure modification. So can I just tweak the molecular structure of a compound to make something new, which I can have intellectual property on, and also where the compound might actually be superior to the original compound. And this is something that is really commonly done in medicinal chemistry, and lots of medicines start as plants. So aspirin, for example, started as a chemical that was derived from willow bark, or from the willow tree, and was modified. So structure modification can be a clever thing to do because you get intellectual property and you also make the compound better than it was before, and possibly improving the physical properties of it, making it more soluble, more absorbable, more deliverable, or sometimes it's because you're making it better able to do what it did before, so better able to activate its receptor, or better tissue distribution. There's many things that you can do to modify a molecule to make it a better drug. There's a lot of this kind of research going on, a lot of structure modification going on for many of the phytocannabinoids, but particularly, as you might imagine, with CBD, because we know CBD works. So can we make CBD better? Is there something that we can do to the structure of CBD to make it better? And this is just an example of some of the many structure modifications that have been done to CBD, where something has been added on, a side chain or some kind of fluorination, or something has been done, a methyl ether group added on, lots of different ways of slightly modifying it and coming up with a new molecule which looks very like CBD but has a minor modification. And so there are many of those out. None of them are yet in clinical development, but many of them have been shown to have very good pharmacological properties or physiological properties, and in some cases work better than the parent compound. So some of these molecules have been shown to be more efficacious in a disease model than the original CBD structure molecule. And finally, in terms of the, what are people doing in the phytocannabinoid space, one of the other strategies is just looking at some of the many other minor phytocannabinoids that are coming from the plant, or even phytochemicals, so not just the cannabinoids, but the terpenes and the flavonoids and all of the other chemicals that come from the plant. Can we make medicines based on the knowledge of what these do preclinically? And I've mentioned throughout the talks that I've given about how, at least in the preclinical literature, we're learning that many of the minor phytocannabinoids have some of the same properties as THC and CBD. So there is scope for drug discovery research in this area. So there's not many of them that have made it yet to clinical development. So CBDV has been pursued by GW in epilepsy, although that trial was discontinued because actually it was found that CBDV didn't work in their epilepsy trial, so they stopped that trial. But they are still actively pursuing CBDV in autism, so that is still an active area of research. And another company are pursuing CBDV in Prader-Willi syndrome, which I've mentioned already, is that disorder where people have insatiable appetites. THCV, I mentioned when we were in the gastrointestinal talk, because it is a CB1 receptor blocker, it has been pursued as a potential medicine for the treatment of diabetes, type 2 diabetes, or metabolic disorders and obesity, and also fatty liver disease. So THCV has been in clinical development, although is not currently in the pipeline of GWs. So many of the other phytocannabinoids, CBG and the acidic versions of CBD and THC, have very good evidence to suggest that they could have therapeutic potential, but as yet they have not reached clinical development. The next strategy to consider is modulation of the endocannabinoid system and how drug companies are looking to pursue this. I wanted to start by saying that you can give an endocannabinoid rather than trying to just increase our local endogenous cannabinoid levels. You can administer a cannabinoid, although to date that's only been done for palmitoyl ethanolamide, or PEA. And FSD Pharma have an ultramicronized version of PEA that they are pursuing in the treatment of the inflammatory effects of COVID, and possibly then moving into other inflammatory areas such as osteoarthritis and endometriosis. And I've already mentioned in previous lectures that PEA has been shown to be beneficial in inflammatory pain in clinical trials and also in depression. So we have a good evidence base that PEA administration is effective in humans. So then the other strategy, the more common strategy, is to try and increase endocannabinoid tone. And endocannabinoids are normally transported. Firstly, they have to get transported across the cell membrane, and then they need to be transported within the cell to their destinations. So there are inhibitors of that membrane transporter for which there is preclinical evidence that they have therapeutic benefit. So if you inhibit the transport of endocannabinoids from the extracellular space into the intracellular space, then you keep concentrations of endocannabinoids high on the extracellular space where the binding site for the CB1 and CB2 receptor is. So that can lead to CB1, CB2 mediated actions that is beneficial in lots of different disorders. But at the moment, there are no membrane transport inhibitors that are currently in clinical development. So then once you get in the cell, because endocannabinoids are fatty molecules, they need to be transported around the cell. And they are done so by these molecules called fatty acid binding proteins. Now, fatty acid binding, inhibition of fatty acid binding protein 5 has been shown to increase anandamide levels and also OEA and PEA levels, because it stops these compounds from being brought to fatty acid amide hydrolase for their enzymatic degradation. So it increases endocannabinoid tone. And Artello Biosciences have an FABP5 inhibitor inhibitor that they have intellectual property on, which has been developed in a number of different areas. So in tumour models, it's been found that these inhibitors stop tumour progression and metastases, and also in inflammatory pain models such as osteoarthritis, they're very effective. So this is still preclinical evidence with an FABP5 inhibitor, but is part of a drug discovery development with a traditional pharmaceutical company, Artelo Biosciences. And then staying with modulation of the endocannabinoid system, there are a number of different enzymes that break down endocannabinoids. And so companies have been looking to develop inhibitors of these enzymes to increase anandamide and OEA and PEA levels. So looking first at Fa, Fa is the enzyme that traditionally breaks down anandamide, but also OEA and PEA. And there are a number of different companies that have been pursuing Fa inhibitors. So Janssen have one that was being pursued in depressive disorders, and Pfizer have a compound that was being pursued in pain and sleep disorders. Now, work in this area really took a significant impact several years ago when there was a company called Bial, who are a French company, and they had developed a Fa inhibitor. And when they went into man, so into phase 1, remember I said that this is the riskiest of all the clinical phases of research. So they went into man and they did a phase 1 study in 6 people, and of those 6 people, 5 people ended up being seriously ill and requiring intensive care, and 1 person died. So this significantly impacted the development of Fa inhibitors. Subsequent research has identified that the particular compound that Bial used had off-target side effects. So it wasn't just inhibiting Fa, and the reason it was so detrimental was not because of Fa inhibition, but because some of the off-target side effects of this drug. So other companies, the Pfizer and the Janssen compounds, have been shown to be Fa selective and they have been in man and haven't had this impact. However, the results of the Bial study, which was very well publicized at the time, really halted all research in this area, and companies like Pfizer and Janssen stopped their clinical trials with their compounds, and really a lot of that work is still on the shelf now because of the tarnishing from the Bial study. There's another enzyme called Na, which specifically breaks down palmitoyl ethanolamide, and so if you inhibit this enzyme, then you get a specific increase in PEA, and we know, as I've said already, that PEA is beneficial clinically for inflammatory pain conditions and many other conditions as well. And there is at least one Na inhibitor which is being developed by Paphos Pharma, still in preclinical development, but it is in their drug discovery pipeline, and it is being used for the treatment of ulcerative colitis and Crohn's disease, so in inflammatory bowel disease, because we know quite a lot of preclinical evidence that PEA is effective in IBD. And then there are MAGL inhibitors. So the MAGL enzyme particularly breaks down 2-AG, which is one of the main endocannabinoids that acts at CB1 and CB2. And there are at least 2 MAGL inhibitors from Janssen and Abide that are in development for mood, pain, inflammation, and Tourette syndrome. So these are all synthetic molecules, and their strategy is all the same, which is in order to try and increase in endocannabinoids and let those molecules activate the receptors to bring about therapeutic benefit. So let's look now at some synthetic molecules that are designed to activate or block the CB1 and the CB2 receptors. So first looking at CB1 agonists, so CB1 activators. This was a drug discovery pipeline within a number of the pharma companies in the early noughties, who developed full agonists of the CB1 receptor, thinking that these would be very good analgesics because THC is an analgesic. But actually what they found when they got into clinical development was that couple of things. Firstly, full agonism of the CB1 receptor can lead to a side effect profile, which is not very tolerable. And the second of which is that they weren't always very effective. Remember that THC does more than just activate CB1. It has many other molecular targets that bring about the analgesic effects. So just singling out the CB1 receptor didn't appear to lead to positive results in trials with these full CB1 receptor synthetic agonists. So they are not being pursued at the moment. But what is being pursued more is this concept of peripherally restricted molecules, so ones that can't cross the blood-brain barrier, so they don't get into the brain, which means that they don't have the side effects that you get from central CB1 activation. There are at least 2 of these compounds. So Artello Biosciences have a compound which is a peripherally restricted CB1, CB2 agonist, and they are developing this as an appetite stimulator, and they are just about to start clinical work with this compound. It's a compound that was originally developed by AstraZeneca and has been in man before, so they can go straight to phase 2 studies with this, looking at its ability to modulate cancer cachexia. And there is another molecule called SAB378, which is a peripherally restricted CB1 agonist, and it's being pursued for the treatment of HIV-associated pain. And then what about the blockers? So in some situations, in some pathological situations, inhibition of the CB1 receptor is a good thing. And one example of that is in obesity and metabolic disorders, where we know that CB1 blockade can reduce appetite, reduce food intake, and improve metabolic symptoms. But the first generation of these compounds, rimonabant, was, although it was successful clinically, it was withdrawn from the market because of its CNS side effects. So people thought, could we still target the CB1 receptor, but without the side effects? Before I say that, rimonabant is still being investigated for some indications. So it's not completely gone off clinical research, but looking at areas where maybe CNS side effects of depression may not be as serious, because people who have obesity often have depression as a comorbidity. So rimonabant is maybe being looked at some other areas, such as alcohol consumption and cigarette smoking, because it seems to help with the addictive properties, to help reduce the addictive properties of other substances. And what's being looked at now in a second generation of CB1 receptor blockers is whether or not, if we peripherally restrict them, again, so keeping them out of the brain, whether you can reduce the side effect profile of blocking CB1 receptor in the brain. And so there are a couple of companies, Goldfinch and Birdrock, who have developed peripherally restricted CB1 antagonists, and these are for a variety of diseases, obviously all peripheral diseases, because it's not going to get into the brain, so there's nothing kind of neurological that you could do with these compounds. So it's kidney diseases, metabolic disorders, inflammatory diseases, fatty livers, and diabetic kidney disease are the types of areas in which it's become well known that overactivation of the CB1 receptor in these disorders is negative, and therefore blocking the receptor would be a positive thing.